specializing in sarcoma at Stanford University Medical Center. He's interested in translational research using information generated from genome-wide gene expression profiling to discover and develop novel markers for the diagnosis and prognosis of human tumors. And his laboratory has been especially interested in the role of macrophages in the tumor environment in carcinomas and in soft tissue tumors. I'm actually, as a junior faculty, I'm particularly impressed and happy that Dr. Van Rijn has also included his a postdoctoral fellow who led the work that he's going to present today. So we're also going to hear from Magda Matusiak, who thank you. Matusiak, who's a postdoctoral fellow in the Department of Pathology at Stanford University and studies spatial transcriptomics, single-cell RNA sequencing, pathology, and cancer immunology. So she is the first author on the paper that's currently on bioRxiv on myeloid diversity in breast and colorectal cancer. And just a couple comments that Matt Van Rijn and Dr. West, who are Magna's mentors, indicated that Magna really did lead this project, including developing the hypotheses, securing funding for the project, and at crucial moments in the project, for example, when they realized that RNA-seq on laser captured microdissection material did not give them the kind of data they were hoping for, she was the one who proposed changes and directions that ultimately led them to the data that went into this manuscript. And I just want to give a plug for Magna that she's on the job market and is interested in continuing this line of investigation. So just two comments, well, I guess 3 comments about the Myeloid Network for 2023. In January, we're going to hear from Alberto Mantovani on macrophage diversity and plasticity in cellular and humoral innate immunity. In February, we'll hear from Dr. Karen de Visser, and then save the date for our 2nd annual full-day symposium in March. Prior to the holidays, we'll send out an email with the call for abstracts so that we can select some abstracts for short talks in the March symposium. So with that, I'll hand it over to you, Matt. Welcome and thank you, and Magna, so much for being here to present your work.
All right. Hello, everybody, and thanks again for inviting us to give this talk. I'll talk for the first half, and then Magda will take over, and you can decide for yourself who you like more. The first slide, I'm a surgical pathologist, so if you don't mind, I will show you a little bit of surgical pathology first. This on the left panel, you see actually, and can you guys see my hand here, my indicator? I guess so. You see a squamous cell carcinoma that is growing to the right and evokes in the surrounding tissue here a dramatic lymphocytic response. So this is the tumor microenvironment at the edge of the tumor at the invasive front. In the right panel, you see a thyroid carcinoma that is growing to the left and evokes a much more subdued response, as you can see here. And so one thing that you can think about is, okay, these are 2 patients and one just has a very strong lymphocytic response and the other does not. However, in this very cruel experiment of nature, this happens to be an unfortunate patient who had both cancers at the same time, a squamous cell carcinoma growing from the left and a thyroid carcinoma growing from the right towards each other here and invoking these vastly different responses in the tumor microenvironment. And so it really shows you the interaction of tumors with their environment is driven by tumors, and that there's heterogeneity there. And other people, of course, had shown this before, but I thought I'd just show you this dramatic case. Now, we have been focusing more on macrophages, and the reason that we did, one of the reasons that we did is that we had been working on a tumor called tenosynovial giant cell tumor. Most of you probably haven't heard about this, but it's a destructive lesion in the joints. It doesn't metastasize, but it can destroy the joints and really impair people's life in a negative manner. And we identified in this tumor a translocation involving the CSF1 gene, shown here in the top left, that leads to very high production of CSF1. And in this panel, you can actually see the break apart where green and red probes are separated when the CSF1 breaks apart on one chromosome, and the other chromosome is here as control. What happens then is because of the very high production of CSF1, the tumor attracts and proliferates a large number of macrophages that actually form the majority of the cells in the tumor. Just like in Hodgkin's disease, where Reed-Sternberg cells are quite rare and the mass of the tumor is made by other cells, the same thing happens here in tenosynovial giant cell tumor. And we've just published a paper together with Magda also in clinical cancer research, if you're interested in this. But based on this finding that we made a long time ago, pharmaceutical companies decided to try out their anti-CSF1 drugs on this tumor because the mechanism appeared to be so clear. And that is shown in the next slide, left panel, where you can see that anti-CSF1 reagents, be it antibodies or small molecule inhibitors, actually led to very successful trials. There were several where these tumors actually become much smaller. They don't disappear entirely, but they become much smaller. And so this was a great success. There have been a number of trials like this, and it's now an approved treatment for this disease. Now, as you know, several studies have appeared where then subsequently, more or less, cancers were being treated with anti-CSF1 reagents. And there actually, the response there was much less overwhelming, much less impressive. And we believe, and I think other people believe that too, that is because in tumors, there reside populations of macrophages with different functions. And so if you inhibit both the pro and anti-tumor macrophages at the same time, your response is not going to be very significant. So a lot of people have worked on heterogeneous phenotypes of macrophages in human and mice, and a lot of work has been done. But a number of questions remain, of course, and that is which populations are distinct, which markers are associated with pro versus anti-tumor activity. What about the presence of these subtypes in various tumor types, and what are their biological functions? And actually, if you want to look at this in a large scale, and if you want to look at tissue microarrays with hundreds or thousands of human tumor samples, it really requires paraffin-embedded, formalin-fixed, paraffin-embedded compatible antibodies that then can allow you to recognize these distinct populations in human cancer. And at the moment, of course, as you know, very few markers exist. CD68, 163, and 209 really react with very large subsets of macrophages and don't provide the granularity that you would like. So our goal was to identify novel markers for human macrophages subsets, and we decided to look at colon and breast. And the question is, of course, why colon and breast? Well, Rob West, my colleague, is doing a lot of work on breast, but also the number of macrophages in breast carcinoma correlate with poor outcome, and that it is the same in many other carcinomas. Colon is an exception in that in colon cancers, if you have a lot of macrophages in the tumor, that is actually associated with good outcome. And so our maybe simplistic hypothesis was that if we would compare macrophages from these two tumor types, that this would facilitate the identification of markers associated with pro versus anti-tumor activity. Now, as was mentioned earlier already, we initially did laser capture microdissection, followed by gene expression profiling on paraffin-embedded material tissue sections. And we really didn't get any satisfying results. And it's really Magda who came up with the idea to really approach this in a novel way, to start with macrophage signatures from published papers and analyze those combined groups of macrophages to look for macrophage subsets, and then look with novel markers in breast and colon cancer to see what we would find. So there's a lot of data in the paper that we have uploaded to bioRxiv, And I'm going to talk about a small aspect of this that will focus mainly on IL-4I1 tumor-associated macrophages or tissue-resident macrophages if it's in normal tissue. Magda will talk more about other parts, but I will also show a bit about normal breast, and then focusing again on IL-4I1, I will talk about the presence of these markers, of these macrophages in tumor tissue. So again, this is a brief summary of how the project developed. There were 4 papers of single-cell RNA-seq data that Magda used for this, 2 from colon, 2 from breast. She isolated all the macrophages from these papers, the information on all the macrophages, and integrated the data that led then to macrophage discovery. And then we were trying to, of course, associate this with biological function, and we hope to convince you of that, and have shown some clinical relevance associations as well. Here, the same project again, just to show you, these are the 11 subtypes that Magda could identify in the single-cell RNA data, colored here differently. She did that using a total of almost 19,000 cells. We looked at 36 breast cancers and colon cancers and analyzed markers derived from these subsets on paraffin tissue sections from these tumors. We subsequently then analyzed it with multiplex IHC, and I will talk mostly about data derived from this approach, and then Magda will talk about the data arrived from the approach for which we used Codex imaging that shows the interaction of these macrophage subsets with non-myeloid cell types. So Magda identified 6 markers that for which she could find antibodies that worked well in paraffin-embedded tissue sections, and they're listed here. And as you can see, these 6 markers identify 5 of the 11 clusters that she saw on the single-cell RNA data. So NLRP3, SPP1 is bidicognate antibodies. IL-4I1 reacts with 3 of the clusters, right? These 2 clusters, ISG115 and CXCL9, react only with IL-4I1, whereas there's co-expression on SPP1 TAMs. And then she further found that the previously described folate receptor 2 subtype could actually be split into 2 groups, one that is positive also for LIVE1 and MARCO, and one that is negative for LIVE1 and MARCO, and only positive for folate receptor 2. The panel on the right shows you that these markers really react with macrophages. They're not 100% specific for macrophages. So LIVE1, for example, also stains endothelium, but that is easily distinguished by looking at the slides and histologic interpretation. And so you can see that all these 6 markers here co-stain CD68 positive cells. So this is again this sort of like vignette. And let's now look at some of the actual data. So here's a schematic of normal colon. This is the lumen of the colon. Here's the lamina propria. There's the lining epithelium. And here's the submucosa with the muscularis mucosa and the submucosa with the blood vessels and whatnot. This is shown here in a real image, 3-color staining. You see here the crypt, okay? This is where the stem cells live at the base of the crypt. As the cells go up the crypt, they mature and ultimately die. And here is the submucosa. And you can see that there's a remarkable distinction in between macrophage types just in this very small region of the colonic mucosa. And I don't have a slide to show you here, but if you stain this with CD68, you just see macrophages everywhere. You don't see any distinction. But here you see a remarkable localization of IL-4I1 positive cells at the top of the crypt in panel 2, I should say, you see these are macrophages that are in the middle of the crypt towards the bottom. They stain for folate receptor 2 and a little bit for IL-4/I1, but nowhere near as much as in the first panel. And then in the bottom of the submucosa here, you can see that you there find folate receptor 2 and LIFE1 co-expression. This right panel essentially repeats the data for folate receptor 2 and LIFE1, but adds in a third marker, MARCO, which is also expressed here in the submucosa, but not in the middle part of the mucosa, and certainly not at the top. And so that is, we thought that was an amazingly beautiful, I still think it's a beautiful image when we first saw it. In the breast, there is kind of the same thing going on. Now, in the breast, you don't have a lumen, of course, right? So there, but you do have breast lobules here, highlighted by this dotted line, that are nested in stroma. And if you, in your mind, think that, you know, this is more like the submucosa than this is, which I think it is, right? And this is more like the lamina propria, if you will, of the breast lobules. You can see that that same distinction is present. The macrophages within the breast lobules are predominantly folate receptor 2 positive, but negative for LIFE1 and MARCO. There's some weak expression, but by and large, they're negative, whereas they're very strong co-expression as you get further away from the epithelium. And there's both co-expression of LIFE1 with MARCO and folate receptor. So it sort of really mimics what we see in the colon and suggests that these are entities that for which the programming is maintained between different tissues. We, of course, next looked at colon cancer and breast cancer. And here's an example of colon cancer. It's growing here from the bottom on up, right? It's highlighted here by this dotted line that shows you the cancer, but also the invasive front, right? The immediate surrounding of the cancer here. And then further away is the tissue that the tumor invades into. And again, there you see a similar thing to what we found in the top of the crypt in the colon mucosa. You see that associated with the cancer cells, there are a lot of IL-4/I1 positive cells that are negative for folate receptor 2, and the opposite is true when you look further away. I'm sorry, I gotta turn this thing off, but I'm on clinical service, so I'm getting cold all the time. So here's a schematic of this core where you see tumor here, IL-4/I1 positive cells are highlighted here in red, the surrounding stroma in yellow. And Magda next computed the distance for each individual macrophage to its closest tumor cell for IL-4/I1 and folate receptor 2, and found that indeed, if you look at this in a single core, you find this remarkable difference confirmed by, confirming what we just see by eye as well. So she next went from this single core where there were about, 500, 600 cells present, to look at many cases of breast and colon cancer to compute the distances there. She looked at a total of 36,000 cells and again found this difference in the distance between folate receptor 2 macrophages and IL-4/I1 positive macrophages. So we next asked, what else can we find, what else can we say about these IL-4/I1 cells in the colon? And we knew that other people had published this, that fragments of dying colon cells at the tip of the crypt can be phagocytosed by macrophages, but there was no marker to identify that particular subset yet. And I think Magda found one in IL-4/I1, and that's shown here. You got here the cancer, or not the cancer, the normal mucosa highlighted by in teal by a PAN-CK marker, And you see that in the lamina propria, there are little dots of this keratin material, and that are present. And like I said, that had been described before. Magda could definitively show that these little fragments of keratin material were actually present in the cytoplasm of IL-4I1 positive cells. So here in the lower left panel of the magnified area, you see an IL-4I1 positive cell, and you see indeed in its cytoplasm are fragments of this keratin material. So the IL-4I1 positive cells are associated with phagocytosis or efferocytosis, however you want to call it. And what I want to point out is that these are individual cells. Okay, these are individual cells that die and that get eaten up by macrophages. And that's important to remember because Magda later on will talk more about areas of necrosis. You know, it's like a larger area of necrosis, and that is associated with a different type of macrophage, and she'll get into that. Okay, so that's this. Now, these macrophages that eat up material have been described also as tingible body macrophages. And the next slide will show you an H&E stain on that, so you can see what they look like if you don't know. But tingible body macrophages are macrophages that have an enlarged cytoplasm in which there are fragments of karyorectic debris from nuclei that have undergone pyknosis and then karyorexics and then are being phagocytosed by macrophages. And these tingible body macrophages, they live in the germinal center in lymph nodes. And that's highlighted in this image by the dotted line. This is the germinal center. And then the remainder of the lymph node is in the surrounding area. And you can see that this is really enriched for single cells stating very strongly for IL-4I1 and only minimally for folate receptor 2. Whereas in the surrounding lymph node tissue, you see folate receptor 2 positive cells with some IL-4I1 expression, but really nothing in comparison to the top one. The next slide shows you here in the top left panel a hyperplastic lymph node. This is just an H&E stain, and here you can see these tingible body macrophages. You see this is the nucleus of the macrophage, and then these little fragments of dark blue material, that's the karyorectic debris that I was talking about, and they are highlighted very well with IL-4I1. Burkitt's lymphoma is a malignant, very malignant tumor with a very high mitotic activity and a very high cell turnover. And so there are always a lot of macrophages associated with these cells. They're scattered through the tumor and they give rise to this, what is called the so-called starry sky pattern, if you're a bit poetically inclined. And you see here that these are macrophages. These are the tumor cells, right? In all the blue cells in between. The arrows point to the macrophages with tingible body particles in them. Again, they are highlighted by IL-4I1. So this whole idea of phagocytosing dying cells is now seen. We've now shown this to you in several different areas. And of course, a question was, does this occur in cancer as well? And the answer is yes. Here you have the invasive front in the left panel of a colon cancer. The colon is growing towards the bottom of the image. And if you look carefully, you see that there are, just like in the top of the crypt of normal colon, there are small fragments of keratin debris that are also taken up by these macrophages, showing here, you know, you've got the little keratin fragment in the right lower panel surrounded by cytoplasm of an IL-4I1 positive macrophage. So these are morphological interpretations, and not everybody is always convinced by them. For pathologists, they are very, very convincing, but we realize, of course, that not everybody attaches the same importance to that. So Magda looked in the gene expression profiles of these single cells and of these subsets for evidence of involvement in phagocytosis. And now, as you remember, the IL-4I1 antibody reacts with three separate subsets or clusters in the single cell analysis. And those include SPP1, ISG15, and CXCL9. Okay, for one of those, CXCL9, the GSEA analysis showed a remarkable correlation with expression of programs involved in phagocytomes, lysosomal activity, and endocytosis. And so that is really I think, a confirmation of what we see by histology, that this is also correlated with the expression profile of these cells in single cell analysis. Now, could this have an effect at all, any clinical relevance? And that's shown in the next couple of slides, where we're just starting to touch on this. And could there be some correlation with clinical situations? Well, we think there is. So if you look at this box here, highlighted here are the reactivity of the different macrophage subsets for IL4I1, or the level of expression of messenger RNA for IL4I1. And as you can see, this group also expresses SERP1 alpha and CD274. Okay, so SERP1 alpha, as you all know, is the receptor on macrophages for the CD47 don't eat me molecule that is expressed on many cancers and is now, a source of some research for therapeutic activity in clinical trials. And then CD274, of course, is involved in the T lymphocyte immune checkpoint programs. Okay, so there is a fairly significant level of expression for these two markers in the group of subsets that's identified by IL4I1. And the question is it possible that these anti-PD-L1 or PD-1 therapies and the anti-CD4 therapies, could they primarily involve the IL4I1 tumor-associated macrophages? And could the presence of these cells, could that actually correlate with response to these therapies? And we do not have that for CD47, but we do have some at least suggestive data for patients treated with pembrolizumab, which is an anti-PD-1. And in the paper by Basset, there were two groups of patients, okay? Those that responded to therapy, with R, right? And the people that did not respond, NR, no response, all right? And for these patients, there were both pre-treatment and on-treatment samples available. And when Magda looked at the subtype distribution in the responders versus the non-responders, she found that in both pre-treatment samples and samples obtained from patients on treatment, there was a relative predominance of IL4I1 positive macrophages, right? And so that really suggests that if you have a lot of these IL4I1 tumor-associated macrophages, you're more likely to respond to pembrolizumab. And this is just the first of analysis that we do and hope to, of course, pursue this further. So to summarize the part of the talk that I will give, and that is focused essentially on IL4I1, tissue-resonant macrophages at the top of colon crypts and tingible node macrophages in lymph node and Burkitt's lymphoma are IL4I1 positive. And we've shown that they are associated in the colon with phagocytosis of fragments of keratin material, which we believe are derived from the epithelial cells. The tingible body macrophages we've identified by morphology, right? You can just see what they are. And in these locations, therefore, they appear to ingest dying or apoptotic individual cells. In cancers, IL4I1 tumor-associated macrophages tightly surround and infiltrate tumor nests, so they are really present within the tumor and at the invasive front, and they contain apoptotic cell bodies derived from individual tumor cells. These IL4 I1 TAMs express SERP1 alpha and CD274, and they are enriched in patients that respond to pembrolizumab. And that is the conclusion of what I was going to say, and Magda is going to take over from here, but I'll drive the slides. Magda?
Great, thanks, Matt. Matt was telling you about how we discriminated the distinct macrophage subtypes in the human tissue, and how we found that they are spatially segregated in breast and colon. And I'm going to be telling you more about this part. So basically how we characterized the cellular composition of those distinct spatially segregated macrophage niches. Please, next slide, please. How we achieved this is by using CODEX. CODEX is a multiplex immunohistochemistry, and we performed this assay in a collaboration with the lab at Stanford. In this assay, we used both breast and colon cancer cases, 24 regions from both of them, 14 cases of breast and 17 cases of colon cancer. This was really cool for us because it gave us an opportunity to profile the distinct macrophage populations in the context of all cells present in the tissue, with an exception of neurons that we did not include. And this was really unique because with this approach, we were able to discover novel and surprising spatial interactions of cells. And we could also map them in the tumor or like in space. And I want to tell you about the interaction of the SPP1 and neutrophils on the next slide. And so interestingly, not only we found that the SPP1 tumor-associated macrophages on the right panel were spatially associated with the neutrophils, but we also found, using immunohistochemistry and immunofluorescence, that the NLRP3 tumor-associated macrophages were also associated with the neutrophil tissue infiltration that's on the left panel. One difference that we could notice right away was that actually I need the previous slide, Matt. Thank you. So one difference we could notice right away was that the morphology of the neutrophils associated with the NLRP3 tumor-associated macrophages on the left is different from the SPP1 tumor-associated macrophages on the right. And the difference is that the NLRP3s were associated with alive neutrophils, as you can, and they're pointed out by the arrows on the image, and you can distinguish them by the multilobular nuclei. But on the right, the SPP1 tumor-associated macrophages were enriched in areas of tumor necrosis. So this is in distinction with this individual cell death that Matt was referring to before. He was saying that the individual cell death and dead cell phagocytosis is likely performed by the IL-4A1 tumor-associated macrophages or tissue-resident macrophages, depending on the tissue. But here, the SPP1 macrophages are associated or enriched specifically in areas of big areas full of the dead cells. So this is basically dying tumor that overgrew its blood supply. And this is mixed up with neutrophils and macrophages. So further, Matt, if I can ask you to change the slide. Thanks. So we already saw this morphological distinction in how the tissue was looking. So the next question was, can we see any differences in gene expression between the NLRP3 and SPP1? Can analyzing these differences point us to give us some hint to understand what's the different function of the two? So we did that, and what we saw on the left panel, we can see the results of the differential gene expression. This is single-cell RNA-seq, and we're comparing NLRP3 terms to the SPP1 terms. And so what we can see is that the NLRP3 terms are enriched in cytokines and that are known to be attracting neutrophils. Also, you can see on the heat map on the right that NLRP3 terms are specifically enriched in IL-1 beta, 6EL1, 2, 3, and 8, which are potent neutrophil chemoattractants. So this suggests that perhaps the NLRP3 tumor-associated macrophages in the tumor are responsible for initiation of acute inflammation, so by recruiting the neutrophils to the tissue, as opposed to the SPP1 that are enriched in lipid metabolism, genes involved in lipid metabolism and phagocytosis. So that suggests that perhaps they have a role in cleaning up the necrotic material that they are enriched in spatially. And next, what we did is we to strengthen this connection and actually quantify the association of the SPP1 with tumor necrosis, is we used a publicly available sample from the 10X. So this is a museum sample, spatial transcriptomics performed on the FFP human breast cancer. And this is a special case and useful for us because there was a lot of necrotic tumor present in that sample. So the tumor necrosis is colored or annotated by orange, and the live tumor is indicated in tail, and the stroma is indicated with colored by gray. And so what we could show that indeed the SPP1 expression is enriched in the necrotic areas of the tumor, as opposed to the, in comparison to NLRP3 that was really interspread in the tumor stroma. So really strengthening this connection, specific spatial association of the SPP1 tumor-associated macrophages with necrosis. And then interestingly next, what we found is, yeah, this is great, is that not only the SPP1 tumor-associated macrophages are enriched in the necrotic areas of the tumor, but also in the hypoxic areas of the tumor. And so this emerged also from the spatial enrichment analysis of the Codex single-cell data. So basically what we found is that SPP1 tumor-associated macrophages are enriched in space with cancer cells that were hypoxic. So we used CE9 marker for the hypoxic to indicate the hypoxia. It's a hypoxia marker. So what you can see now on the right is a zoomed in fragment of a colon cancer invasive front that we imaged with Codex. And what you can see there is that the cancer cells indicated with pancytokeratin in white or gray that are hypoxic, indicated by CA9 with pink, are surrounded by SPP1 tumor-associated macrophages indicated in orange. So this, together with the association, spatial association with the necrosis, indicates that the SPP1 macrophages have something to do perhaps with the cleaning up of the necrotic areas in the tumor. So now coming back to the SPP1, no, sorry, to the NLRP3 macrophages, I told you that that the single cell data indicated that the NLRP3s might be recruiting the neutrophils through secretion of the 6TL1, 2, and 8, as well as IL-1 beta. However, we know that just production of IL-1 beta is not enough for the IL-1 beta to be biologically active, because IL-1 beta is produced as a proform in the cell, and it has to be proteolytically cleaved to be and secreted to be biologically active. And that cleavage is performed by inflammasomes in the cell. So interestingly, NLRP3 is a receptor that when activated, it's initiating inflammasome activation. So that complex that is necessary to cleave and activate IL-1 beta. And then when IL-1 beta is secreted from the cell and is biologically active, one of its roles is to recruit the neutrophils. So interestingly, when we looked at the distribution of the NLRP3 protein in the tissue, next slide, please, Matt. What we could observe is that we can distinguish two distinct distribution of the NLRP3 protein in the cell. So now I'm talking about the middle panel that is showing the diffuse distribution of the NLRP3 in the CD68 positive cell, which is a macrophage. But we could also we could also find areas that displayed NLRP3 aggregation in one spot in a macrophage that is also labeled by CD68. And I forgot to tell you that when inflammasomes get activated, all the proteins that form inflammasome become aggregated in one spot that is perinuclear. And actually the spec formation, or this aggregation of the proteins, is used as a readout for inflammasome activation. And usually people use ASC, but we couldn't optimize the ASC for the FFP, so we're using NLRP3. So what I'm trying to say is that we are interpreting this NLRP3 aggregation in a cell as a indication or a proof that NLRP3 inflammasome is activated in that cells. So now, interestingly, what we found is that the NLRP3 cells with the specs, so the aggregated form of the NLRP3, were spatially associated with neutrophils, but the regions with diffuse NLRP3 expression was not. So we computed that. We can see that on the next slide. That when we counted the neutrophils in the tissue and compared the areas with diffuse morphology or expression of the NLRP3 to aggregated NLRP3 in a spec that we interpret as inflammasome activation, we could see that indeed the inflammasome activation is likely associated with this with the neutrophil infiltration in the tissue. So this suggests that inflammasome, and actually this is the first histological demonstration that inflammasome activation in human tumor is happening, and that it is associated with neutrophil infiltration. So we think that that this event is shaping the tumor immunity and is shaping the tumor niche, and this resulting in acute inflammation because of the neutrophil infiltration. And so that was very cool and encouraging. To kind of validate that, and we looked in another indication. So on the next slide, we examined Crohn's disease, and we looked into Crohn's disease because we knew that that in Crohn's disease there are areas of the tissue that are heavily infiltrated by neutrophils. So what we wanted to ask is whether the NLRP3 inflammasome might be involved in that too. So what we did is on the left panel, we looked into the areas of Crohn's disease that are heavily infiltrated by neutrophils. And now on the right immunofluorescence panel, what you can see is that we indeed found that the areas that are heavily infiltrated by neutrophils in Crohn's disease are also infiltrated by macrophages with an activated or aggregated NLRP3, which we interpret as activated NLRP3 inflammasome. So that was really cool. And we thought, because as I said, this is the first real demonstration that this is happening in Crohn's disease, because and in cancer in human, because the previous most convincing data coming from the human that was indicating inflammasome involvement in cancer and Crohn's disease were based on SNP associations, and one clinical trial that was using anakinra, that is targeting IL-1 beta in lung cancer. No, sorry, in arteriosclerosis. And what they found is that IL-1 beta targeting in arteriosclerosis causes lower incidence lung cancer in those patients. So indicating that perhaps inflammasome activation might be involved in tumor genesis in the human. But what we show here is the first demonstration that this is really happening on a histological level in the tumor and in Crohn's disease. So we were really excited because that perhaps might indicate that NLRP3 could be now targeted in those indications. So on the next slide, to kind of further support this and inquire, we wanted to inquire whether there is any prognostic association of the inflammasome activation with outcome. So we collaborated with Aaron Newman and Bogdan Luka from Stanford. So it's a Newman lab. And so what we did is we looked for prognostic associations of gene signatures that were a surrogate for infiltration of the distinct macrophage subtypes, or actually the whole macrophage niches. So understood by a spatial enrichment of the specific cell types together, like for example, NLRP3 and neutrophils, or SPP1 and neutrophils. So we were asking whether there is any association of the abundance of those cell types or the abundance of the niches that we were studying with outcome. And what we found was really cool. And I should say that we included as a reference a gene signature that is most on the right. So it's folate receptor 2, SEPP1, SLC40A1. This gene signature was recently published by Rodrigo Ramos and Julie Heft from Paris. And so what they showed is that this is associated with positive outcome in breast cancer. So we really could recapitulate this data. We saw the same, which really corroborated our approach. But what was really interesting, what we found is that, so I'm going to now refer to the two red arrows. So the right red arrow is showing that the NLRP3 tumor-associated macrophage abundance in breast cancer alone is not associated with outcome. However, the abundance of NLRP3 tumor-associated macrophages and neutrophil niche is associated with better outcome. And so we interpret this data and as so this kind of correlates with our histology. So we showed that the NLRP3 distribution, the diffuse distribution in the macrophages is not associated with neutrophil infiltration in the tissue. However, only after NLRP3 is being activated and aggregated in the cell. This is now associated with shaping of this inflamed tumor niche that contains NLRP3 macrophages and neutrophils. And so we interpret this enrichment of NLRP3 and neutrophils as activated inflammasome. So if it's true that that this signature can really distinguish or show or inquire the inflammasome activation in the tissue, then that would indicate or like support this notion that perhaps NLRP3 could be targeted in tumor because it is associated with bad outcome in breast cancer, but not colon cancer. And I have to say that we also examined the single association of neutrophils with outcome, and we could see also that it correlates negatively with outcome. But we have to also indicate that NLRP3 activation is only one of the reasons or one mechanism that might be shaping or recruiting the neutrophils to the tumor, and likely there are others that we are not looking at. And so with that, I would like to look at the summary slide and tell you how we interpret the big picture. And so here, what we show is that what this work really taught us or showed is that macrophages, different subtypes of macrophages occupy spatially distinct niches or spatially distinct regions in the tissue. And we showed that those distinct regions not only have those are seeded by the different population of macrophages, but they are also characterized by the different cellular composition. And we think also different function. And we showed some of the functions, like, for example, that the IL-4A1 tumor-associated macrophage niche is likely associated with the areas with high cell turnover and high cell death, and the function of NLR and the function of the IL-4A1 macrophages in that niche is to phagocytose or efferocytose, as opposed to NLRP3 that we were also extensively talking about today, and that is. We showed an evidence that the NLRP3 inflammasome is activated in an NLRP3 macrophages in the tumor microenvironment, and likely that is shaping tumor immunity by inducing acute inflammation because the cytokines together with IL-1 beta are likely recruiting the neutrophils to the tissue. But really something that I wanted to say, something that emerges from all this is that we propose that macrophage niche is a fundamental functional building block of the tissue. And this is because we show that the different macrophage niches, functional niches, are not specific to a single organ or disease. They are shared between normal and cancer tissue. We showed that the NLRP3 is shared by the tumor and by inflammatory disease, Crohn's disease. So what we show is rather that the distinct macrophage niches are performing the same function in different organs, in different diseases, and therefore they're we think they're fundamental building blocks of the tissue. With that, I would like to thank you for your attention, and I would love to take your questions and discuss. Sorry, I forgot to, of course, acknowledge all the people that contributed to this work. most of the people are from Stanford. Technical support from our lab. Codex imaging was done by John Hickey and Gulan Lu in the Nolan lab. And really importantly, the survival analysis was performed by Bogdan Luka from the Newman's lab at Stanford. And very, very important, the pathology or the histology was interpreted with help of surgical pathologists at Stanford, Matt, Rob, Ginny Shen, and Greg Charville. Great. So I don't know, Jennifer, do you want me to, or Judith, do you want me to just read the questions, or do you want to read the questions? How do you want to?
We'll read them for you and then ask.
Okay, great, thanks.
Thank you very much for an amazing talk. The images you show are so beautiful and instructive. I would like to ask a couple of questions first. You know, I'm curious as to what you think that the association of neutrophils with these NLRP3 macrophages are doing that leads to better outcome in cancer.
So actually it leads to worst outcome.
Worst outcome. I thought it was positively associated with overall survival.
Yeah. So the folate receptor 2 SLC as CPP1 was positively. Yeah, great. Thanks, Matt. So the question is, what might neutrophil and do to the tumor so that the outcome is negative? So we don't really have any data to definitely say what's happening. But what I can say, the neutrophil infiltration is in general associated with what's also what we show here. It's associated with worst outcome. So I'm not sure what's the mechanism. Perhaps it's because the immune system is overreacting and neutrophils might be activated and it's just overwhelming the response. That's what I think is happening, but I have no data so this is just a speculation.
Thank you. I think I read your diagram like oppositely. So thank you for clarifying that. I do have one more question, and that is, what is the gene signature that allows you to distinguish NLRP3 macrophages plus neutrophils from just NLRP3 positive macrophages? Because this is basically gene expression, right?
Right. This is gene expression signature. So we just so it's in the supplemental figure or the supplemental table, you can find the exact genes, but it's just so it's NLRP3 is going to be NLRP3, 6L128, and I think PLER. And then NLRP3 tumor-associated macrophages with neutrophils is just the same plus the neutrophil signature that we derived from the single cell plus actually informed from the spatial transcriptomics.
Okay, thank you.
You're welcome.
Jennifer, did you have any questions?
I think we can go to the chat. Tatiana, I just saw you turned on your camera. Do you want to ask your question?
Absolutely. We'll be glad to. So, you know, at the first part of the talk, right, when Matt was talking about IL-4, I1 macrophages and their presence predicting a better response to T-cell checkpoint inhibitors, are you suggesting, you know, what would be the data to actually suggest that those are the cells that are reacting to PD-1 inhibition, or they are just setting the microenvironment and the immune system to react to that?
That's a great question. So our data shows that the macrophages that are IL-31 positive are also PD-L1 positive, and the treatment was against PD-1. So we don't have a data to show that it directly affects those IL-31 positive macrophages. We only show that they are predictive, or their abundance in the tumor is predictive of the response.
Did you look at PD-1 itself on those cells?
We did. I think, I'm not sure if this is on the figure. In the single-cell RNA-seq, just the data available, there was just very little expression at all. So it was not conclusive. It's just, so what I'm trying to say is that very little number of cell, like I think 1% of macrophages express, I mean, we can detect by single-cell RNA-seq only that 1% of cells express. So it's not probably what's happening, but this is what we can detect. So we can't really conclude. But what we've been suggesting is that because the IL-4A1 are expressing SIRP1 alpha and PDL1, they might be direct targets for CD47 and PDL1 therapy, right? So we can't really talk about the PD-1 because we couldn't see the expression. But I think this question could be really well addressed by staining of the protein. Yeah.
Thank you.
Yeah, you're welcome. Maybe just to add to this question, there were several papers over the last few years showing that actually the expression of PD-1 and PDL1 both on macrophages are more associated, I'm not sure if you're familiar with that, but are more associated with the response to the PD-1, PDL1 axis checkpoint therapy than the expression of those molecules on T cells or cancer cells. So my feeling is that indeed the effect is driven by the actual direct targeting of macrophages, but we don't have data.
We can go to the next question. Please feel free to turn on your cameras and ask your questions yourselves or raise your hand. The next question is from Alex. What do you think about CXCL9, CXR3 interactions between macrophages, given CXCL9 positive MAC is a subset of the IL-4I1 TAMs and T cells that have been observed in colon and melanoma cancers? I see immune checkpoint inhibitor responses.
So we didn't have a chance to look at that, but I can say that when we are performing a global analysis of the spatial cell interaction in the colon cancer using Codex, we don't see interaction of the IL-4A1, so the CXCL9 positive cells, as being reported with the T cells. What we see is predominantly that the IL-4A1 are present in the epithelial compartment, and the the areas that are heavily infiltrated with the lymphocytes are associated with the folate receptor 2 macrophages or live ones rather than IL-4A1. So I know Florent Ginoux showed this, is just not what we see.
Great, thanks. There's a question from Santiago Acero Bedoya who asks if you could speculate if these IL-4I1 MACs are the same or similar to an IRF8 macrophage that was reported by Ming Li's group that drive exhaustion of CTLs.
I'm not familiar with that paper. Is the IRF8 the population that is ISG15 high? Is that the same population, CXCL9, 10, 11?
Santiago, are you on?
Yes, sorry. I'm not quite sure. I think it just struck me like a lot of similarities. The IRF8, I think the big thing is that IRF8 is also the main transcription factor for classical 1 dendritic cells. And they were showing that they are able to present that antigen. And they show that they're both in normal breast tissue as well as in breast cancer tissues, which is kind of similar to the IL-4I1 that you guys show. And also thinking about the fact that these are phagocytic in your model. Yeah. And so I'm wondering what happens with the antigens or the debris that's taken up by these MACs and how that might be interacting as well with, their tissue and their microenvironment.
Cool. So if you're saying that this cell population is present in normal breast, then likely if they were studying human, it's not that the same population, because we can't see any IL-4A1 positive macrophages in human breasts, in the normal breast. But are you talking about macrophages or dendritic cells?
Macrophages.
Macrophages, yeah. So we definitely don't see any IL-41 positive macrophages in healthy breast.
But they were present in the healthy colon, right?
Yes, they are present in the healthy
Okay.
colon on top of the lamina propria, and we think that is because they are phagocytosing the dying intestinal epithelial cells on the top of the crypt. And we think that they're likely not present in the breast compared to colon is because there's far less cell turnover at steady state in the breast. That's what we're thinking. But you know, if you want to follow up on that, we can just look at the data later. I'll be glad to.
And
yeah.
if I could tack on just a small question to that, which is maybe a low-hanging fruit, but I'm curious how maybe this fits into the idea of like M1, M2 polarization and where the subsets that you've defined maybe fall into that spectrum.
Yeah, that's a great question. You know, it's a hard question because I'm not sure. So, you know, it's just not really what we see, that there's this dichotomy in vivo in human. Like single cell RNA-seq already showed that there's so much heterogeneity. There's not only like 2 spectra, there's so many papers like refuting this, like saying that there's so much more, and we should be really cautious not to fit M1, M2. And so, yeah, I'm trying to be cautious too. What we see is that there is a lot of heterogeneity, and it's not easily like dichotomized. There's just right, and then you know we're just starting to characterize the function, and I think much more functional studies have to follow to really. And also clinical associations to really say whether the subsets that we are discovering are pro or anti-tumorigenic. We're just showing like basic cellular associations and functions, like you know phagocytosis versus plasma cell maintenance in the tissue, stuff like this. So you know we need to follow up with the clinical studies and functional studies to say whether this function is pro or anti-tumorigenic.
So we have a few more questions from Samarth Hegde. You're invited to ask your question in person, but I can start by reading it. Exciting study with such rich insights is the SPP1 osteopontin is frequently expressed in epithelial cells and fibroblasts. So they wanted to know whether this signature that you see in macrophages also included myeloid markers, or how you distinguish that from these other types of cells.
Great, that's a really great question. I should have addressed that while I was speaking, and you can really see how we did that in the paper. But how we did that is we used co-staining with CD68. And in Codex, we have more markers, CD68, CD163, to be 100% sure that the cells that we are analyzing and describing are macrophages. And so I'm talking about immunofluorescence and the Codex. In Visium signature, oh, okay, you're talking about the Visium, sorry. So in Visium, how am I sure? Yeah, I'm not sure if this is coming specifically from the macrophages. Yeah, we just plotted the SPP1. We didn't really co-plot with C68 and stuff like that. Yeah, perhaps this is overconfidence from our side. It's because we just saw it by staining. Yeah, we should do that. Thanks for the suggestion.
Here's another great question from Daniela Sure. So super interesting results. I was wondering if you'd characterize the transcriptomic state of the neutrophils associated with the NLRP3 TAMs compared to neutrophils in other regions of the tumors.
We have not. This is a really cool question. We would love to do that. Yeah, one way of doing this would be using Visium or laser capture. So I guess we even have data that could address this, but we didn't really look at that.
So I have a question based on all of these comments. So that one of the things that we see in tumors when they're responding to therapy is you get T cells that are highly stimulated, and then they invoke their automatic responses to shut down that stimulation. So more PD-1, more checkpoints. And it looks like macrophages do something similar. So I wondered if you have tried to apply your markers to other disease states. I know you're looking at Crohn's disease, but what about infectious diseases?
Oh, cool. Yeah, we have samples lined up. We're so excited about that because, my background is innate immunity, and I was studying salmonella on my PhD. So totally excited about this question. We just have the samples lined up. There's so many things that we would like to check, not only infectious disease, but we also collaborate with head of our department, Tom Montin, that is studying neurodegenerative diseases. So we'll be looking at that. Yeah, I mean, establishing these markers for human tissue FFPE for clinical samples, it's just it's great for us. I can do this for the rest of my life, really. Looking at different diseases, and yeah.
Great. I one more question, and then we can ask the other questions. It wasn't clear whether you've actually done a parallel study of staining tissues and then say single cell sequencing from the same tissue, and that might help help you correlate gene expression signatures then with the actual localization and identification of different types in tumors. Have you done that on
So that's a great suggestion. We didn't do that on the same samples. What we did is we analyzed publicly available single cell RNA sequencing data, and we correlated that with our staining. So, and this was really helpful, like to understand the function, like, for example, from the data I showed you today, when we saw the spatial association with neutrophils with NLRP3s, we asked what can be, what can be mechanism of this, what can be leading to creation of this acutely inflamed neutrophil-rich niche, and looking at the single cell RNA-seq really showed us that the NLRP3 TAMs specifically are enriched in the neutrophil hemoattractant. So so that led to the idea, okay, but they're probably recruiting the neutrophils, and then that then we saw the inflammasome activation, and definitely combining the single cell and correlating that with the histology was very helpful and informative for this study. Yeah.
There's a couple more questions at the end that
Yeah,
are interesting.
Matthew, you still here? Matthew
Yep,
Park? Oh, no, sorry, Matthew
Yep.
Park, last question.
Yes, sorry. Hi. Great talk. I really found it really interesting. I just had a quick question because you did raise an interesting thing about the basic biology for one of these macrophage populations, the IL-4I1s. And because they're macrophages, we presume that they are engaged in, phagocytic uptake, and whether that's efferocytosis or just general uptake, one can ponder. But I was wondering whether you had some data showing that this is a causal thing, where in fact actual uptake induces this program, whether it's just ambient IL-4, for example. I mean, you are calling them IL-4I1 max, so IL-4 may be alarmins. So I was wondering if you have data showing causality, or whether it's possibly coincidence. And it's because you're showing imaging, I was wondering whether you had other markers for the macrophage populations you identified, like the SPP1, whether you stained that with the antigens, and I guess didn't see any of these antigens in those macrophages.
Right, that's a great question. So I think there's two questions. One is that whether we know whether the IL-4I1 is causally connected to phagocytosis or efferocytosis or whatever is happening, whether the IL-41 is coming before or after, is it induced or is it coincidental? So we don't know. It's a great question. We're trying to address that by doing in vitro experiments in a collaboration with phagocytosis lab, CD47 lab, Matt.
The Weizmann lab.
Of course,
The
the
Weizmann
lab. Thank you. So yes, so we're trying to address that with Weizmann, and we don't know yet. This is a great question. So we're on it. And the second question is, whether we showed that the uptake of antigens, so we're not showing antigens, we're just showing that cell body, or that, I don't know if it's that, we're showing the fragments of epithelial cells that are being engulfed and are inside the IL-41s. And the question of Matthew is whether we showed that it is not happening. It's not happening, for example, in folate receptor 2 or the SPP1. Yes. So it's not happening. It's specifically only happening. It's performed by the IL-41 cells, macrophages. But we did not detect these events associated with folate receptor 2 or SPP1 or NLRP3. We didn't detect that. And I should say it's not only epithelial cells that we saw are being, or the fragments of epithelial cells that are being phagocytosed, but also the dying B cells in the lymph nodes, germinal or Burkitt's lymphoma. Does that answer your question, Matthew?
Perfectly. Thank you very much. Very interesting.
Yeah, you're welcome.
I have a question. Have you observed hemoxygenase in any of your analyses of data sets or in your immunostaining, hemoxygenase positive macrophages?
So in this study, we did not. We performed a different study where we used MIB, which is also multiplex immunohistochemistry. We used HO1 antibody, hemoxygenase 1 antibody there. And we did see some HO1 positive macrophages, but in a very specific regions and rather rare. So we didn't really see that a lot. And it can be antibody. I'm not sure what we're supposed to see.
Just in single cell sequencing of experimental tumors, at least, hemoxygenase positive macrophages can be a really major population, and they're also associated with markers of dying cells. So one thing, it's not clear if it's dying macrophages or if they're phagocytosing, and you're seeing a lot of
very interesting.
Yeah, so that would be something interesting.
Okay. Yeah, very interesting. Yeah. Yeah, I can share the antibody, I guess, but if I'm not sure, are you working with human or?
Some.
Yeah. Yeah, super cool question. We didn't really look at this.
Okay. Well, I would say most of the markers you describe are actually seen in mouse signatures too, which is comforting for those of us modeling disease in mice. And I think it shows that spatial understanding is really important too. So it's a really insightful work, and really appreciated your sharing it with us. Unless anybody would like to ask any more questions in person, I think that that would be it for today. I also would like for those of you remaining to introduce Adam Prater and Victoria Hernandez, who are my associates at the Moores Cancer Center at UCSD, who will be taking over administering this seminar series for us, and you'll be hearing from them more next year. So thank you, Adam and Victoria, for being present today and helping out. So thank you again, and we will be sending you the questions in the chat in case you'd like to address them further with people. So
great, fantastic.
Okay,
Yeah,
great.
I want to also encourage you, if anyone wants to connect and discuss further, we will be really glad to do that. Yeah, thanks for having us.
thank you.
Thank you. Happy
Bye.
holidays, everyone.
Hey,
Happy
Jennifer,
holidays.
Bye-bye.
stay on for a minute.